Mr. Chancellor, today we welcome to our campus Professor Martin Chalfie, co-recipient of the 2008 Nobel Prize in Chemistry. Professor Chalfie and his fellow researchers Osamu Shimomura and Roger Sen were awarded the Nobel Prize for their discovery and use of a protein that allows a particular species of jellyfish to glow bright green. Professor Chalfie realized that the protein could serve as a biological marker when listening to a seminar in his department at Columbia University. In the 3 decades since that aha moment, the use of this green fluorescent protein, or GFP as it is called, as a biomarker has enabled the mapping of tens of thousands of different proteins that reside in cells. It's now one of the essential tools in biological and biomedical science research. Using GFP, one can now actually see, using a microscope, what was otherwise invisible. So, what does this reveal? We can, for example, watch nerve cells communicate with one another in real time. Incredible. We can also watch cells divide in a living animal, and we can watch healthy cells replace disease cells in cell therapy research, eventually leading to possible new treatments for diseases such as muscular dystrophy. Shortly after the awarding of the Nobel Prize, Professor Chalfie published an essay about the confluence of good luck, missed connections, making connections, curiosity, and sheer brilliance that led to the GFP breakthrough. This essay included many fascinating images, including antibody staining that revealed receptors in roundworms, and the now famous cover of the journal Science that announced his group's glowing green discovery to the world. But perhaps the most fascinating image in this essay is a simple sheet of paper, a messy, handwritten jumble of names, phone numbers, and cryptic notations. The day after that fateful seminar, Professor Chalfie spent the day on the phone tracking down people who knew more than he did about GFP. One name led to another, then another, then another. He wrote, "In a sense, GFP is a wonderful metaphor for our work as scientists. Just as GFP and other fluorescent molecules absorb light of one wavelength and convert it to the light of a different wavelength, we too take in what others have learned about the world, add our observations and insights, and produce additional gains in human knowledge." Scientific progress, and really the creation of any new knowledge, is not a solitary process. In fact, he devoted part of his Nobel acceptance speech to debunking common scientific myths in the public, including the misconception that scientists work in isolation and solitude. He said, "Each of us has been a step in the very large development of GFP, and we have not been alone." Today, we pay tribute to Professor Martin Chalfie for his spirit of collaboration, his humanitarianism, his scientific curiosity, and for literally illuminating life. Mr. Chancellor, I present to you Martin Chalfie so that you may confer upon him the degree of Doctor of Science, honoris causa.
I will now ask Dr. Chalfie to come and make his address.
Thank you. Chancellor McCall-McBain, Principal and Vice Chancellor Fortier, Mr. Panda, Chair of the Board of Governors, proud families and guests, and most of all, members of the graduating class of 2022, I am greatly honored be asked to help you celebrate this important day. I am, however, hesitant about giving a speech, as I certainly don't feel that I can give advice, the staple of graduation speeches. Several years ago, I was one of 70 adults from various fields in the sciences and the arts that were invited to participate in a 4-day event for 150 amazingly talented and accomplished high school students. Each adult got up and talked about themselves and their interests. But virtually every speaker ended their talk with advice to the students. As I listened to these speeches, I realized that the speakers were not actually talking to the students, who, after all, like you, don't need any advice, or didn't need any advice. They were talking to themselves, or rather their younger selves, saying what they wish they had heard. They were all in effect saying, I made mistakes, I failed, I wish someone had corrected me. Here is the advice I wish I had heard. Of course, this thinking is nonsense, because failure and mistakes are the very beginnings of all we do. And who really listens to advice anyway? When it was my turn, I simply said, as I say to you now, you have been amazing. I don't see that changing, and I'm eager to see how you will amaze us in the future. Thinking about advice, the most interesting advice I ever received came to me in the hours before my PhD thesis defense. Actually, it was a rite of passage. So-called friends prepped me for the exam by giving me sample questions, and they assured me that I was going to be asked one question in particular. If when you started you knew what you know now, how would you have changed your research? They also told me that if I answered the question in the way I probably would have, that is, by saying I should have done something entirely different, then the examiners were sure to say, well, if so, you fail. Go do those experiments and then come back and defend. I was destroyed because I didn't know what to say. I was clearly going to fail. After my friends let me stew for a while, they told me that the question, what would you do differently, could be answered in only one way. And that way was, I would have done it faster. In fact, I was actually never asked that question, but it was just there to scare me at the beginning. And working fast and slow, and usually slow in science, is what I've been doing ever since. I know this is the Science Day convocation, so I'm among friends, and while I'm very glad to share in the enjoyment of your accomplishments so far, my primary emotion today is of envy. For many of you, a life in science is really just starting. You will get to see all the new things that will be discovered and understood. I envy you what you will see and what you will do. While other fields of human endeavor can look back to a golden age or to past greats whose works are still revered and enjoyed, Shakespeare, Mozart, Beethoven, the golden age of science is always right now. We build on past knowledge, most of which becomes information in textbooks, not associated with any names, but we are always in the midst of the greatest era of science. I don't think we need any more proof of this than to look at how fast vaccines were developed to protect people from COVID-19, an astonishing accomplishment that was built on years of basic research. I've been around long enough to have experienced many revolutions in biology. When I was in college, the cutting-edge science was biochemistry, or so I thought when I chose that as my major. It sounded a lot sexier than biology or chemistry. Soon, however, the molecular biological methods were developed to manipulate and sequence DNA, and enormous areas of study opened up. For the first time, a molecular understanding of development and genetics was possible, and biotechnology gave rise to numerous businesses. Research possibilities exploded. I and many other researchers study a small roundworm, a nematode called Caenorhabditis elegans. Enabled by molecular genetic techniques, researchers with this tiny worm have discovered genes needed for cell death, for organismal aging, for cell development, and for multiple levels of gene regulation. More generally, the DNA sequencing that gave us the human genome and the genomes of so many other organisms has greatly increased our understanding of evolution and yielded the bases of many human diseases. Even more importantly, in my mind, we are increasingly looking at organisms and their interacting cells as wholes, so-called systems biology. And the list continues. The development of new imaging methods and tools have changed and continue to change our view of life. The development of gene editing technology is also redefining even what we can study in biology. I was amazed a couple of years ago when I was at a meeting and I heard about people studying the patterning of butterfly scales, the development of emu wings, and the organization of the turtle's shell, all by modifying genes with CRISPR-Cas9. And researchers are mutating genes in lab organisms to mimic defects found in people with rare diseases, to model, understand, and ultimately treat human diseases. And finally, computational methods and machine learning are helping us obtain an even deeper understanding of biology, for example, protein structure. I don't know what the next breakthroughs will be, but I do know that they will be built on an ever-increasing knowledge base of basic research. You will see these breakthroughs, in some cases make these breakthroughs, but most definitely benefit from them. We all need to support these efforts. This support, however, will not be easy. This rosy picture is clouded by antagonism to science and attacks on scientists. People don't like facts and results that don't support their beliefs or agendas. As a result, academic and scientific freedom and the autonomy of universities and national science and medical academies are under threat in many countries. Scholars at Risk recently issued a report, Free to Think 2021, which found that there were 332 attacks on such institutions in 65 countries and territories last year. And in many countries, doctors who complained about governmental failures to contain COVID have been threatened, harassed, and even arrested. Scientists are unfortunately easy targets for politicians. We as scientists and their allies should defend and support our colleagues all over the world. And that phrase, our colleagues all over the world, brings to mind one of the most positive aspects of science and one that allows me to end on a high note. To me, science thrives when a variety of views, experiences, and perspectives are present. Several years ago, I Googled famous scientists, and to my dismay, the first 25 images were all of white scientists, and only 2 of them were of women, Marie Curie and Rosalind Franklin. But when I look at my lab, I see something very different. If I consider the sabbatical visitors, postdocs, graduate students, undergraduates, and technicians who've worked in the lab over the last 10 or so years, I see men and women, all types of people, who have come from Canada, Chile, China, Colombia, Greece, Iceland, Israel, Peru, the Gambia, United States, and Vietnam. Each has added uniquely to the working of the lab, and all have made it a wonderful place for discovery. The increase in opportunities in the sciences for all is one of our most promising trends. We are enhanced by everyone's contribution. As I indicated at the beginning, I am not very good at giving advice, but I do have a wish for all of you graduates, that you enjoy and relish your uniqueness and marvel at the future. You are now about to embark on what a college friend of mine once called your infinite summer vacation. It's an exciting time. Enjoy it, and congratulations.